Hi, everyone. Welcome to this week's worldwide series of the Neurobiology of Addiction seminar series. This week, we're lucky enough to have Dr. Cassandra Gipson joining us to tell us about her recent work. And I will let Anka Chakrabarti do the formal introductions.
Do I wait for a while because people are joining in right now? So we can wait for a minute or two and then start. Right?
That is true. There's usually a bit of an exponential rise in participants between 11 and 11:03. So we can give it another 2 minutes here before we do the introductions. Thanks. And just a reminder for anybody who might be new this week is that all of these talks are recorded. They're also directly live streamed to YouTube. But if you go to the worldwide Neurobiology of Addiction webpage, you can find our archive of all the previous talks in the series with links to watch them on YouTube. And I believe they'll stay up there for the foreseeable future. So there's also a really great resource of previous recorded talks that you could browse. And I think whenever you're ready for the introduction, we can give maximal time to Dr. Gipson if we get started. So people might still be trickling in, but we'll give it a go.
So hi, thank you for joining in today. And so today we have Dr. Gipson with us. Dr. Gipson received her bachelor's degree in psychology from UCSD in 2004 and a PhD in 2010 from the University of Kentucky in psychology department with Dr. Michael Bardo. Dr. Gipson then contributed to a postdoctoral training at the lab of Dr. Peter Kalivas at Medical University of South Carolina, who will also be presenting his work next week here. So she studies how transient synaptic plasticity in the nucleus accumbens contributes to diverse behavior symptoms of addiction. Dr. Gipson joined the Department of Psychology at Arizona State University in 2015 as an assistant professor. She joined the Department of Family and Community Medicine at University of Kentucky as an associate professor in 2020. The focus of her research is to identify novel neurobiological and behavioral mechanisms to guide the treatment of addiction, including glutamatergic, dopaminergic, and neuroimmune and ovarian hormone mechanisms underlying addiction to various drugs of abuse. Dr. Gipson's lab is supported by the National Institute on Drug Abuse, and she recently was awarded the College of Drug Dependence Young Investigator Award. So without further ado, I will give the stage to Dr. Cassie.
And I'll just remind everyone. Oh, sorry. I'll just remind everyone that, as always, we can use the Q&A function. There's no chat, but you can drop your questions in the Q&A function, and we can address those at the end of the seminar. Sorry, Dr. Gipson, I cut you off.
Okay. Thank you all so much for being here and for hosting me. I'm really excited to talk to you today about some recent work that my lab has been doing, focusing on neuroimmune and glutamatergic mechanisms of nicotine addiction. So just to give you a little bit, I don't know if my mouse is working. Oh, there it goes. Just to give you a little bit of an overview, I'm going to give an introduction on tobacco use disorder. I'm going to kind of narrow in a little bit to neural circuitry across drug classes and substance use disorder, and then narrow it a little bit more to neuroimmune mechanisms in substance use disorder. And then I want to talk about some background on glutamate dyshomeostasis in nicotine addiction, focusing specifically on the glutamate transporter and synaptic plasticity within the reward pathway. Then I'm going to talk about some of the work we did a few years ago on the role of NF-kappa-B, or the nuclear factor kappa-B pathway, in nicotine-seeking behavior, focusing on N-acetylcysteine mechanisms of action. And this is a compound being looked at in clinical trials to treat substance use disorders. Then I'm going to talk about our work on nicotine and microglia, showing you some more morphological data and focusing on sex differences. Then I want to switch gears a little bit and talk about neuroimmune signaling as potential treatment for nicotine, focusing on a novel compound called Troraluzol. And then I'm going to talk about some future directions and some other ongoing studies that we have in the lab. Okay, so tobacco use disorder is a leading preventable cause of death, and within just the United States alone, contributes to 480,000 premature deaths in the United States due to tobacco-related causes. Nicotine is generally considered to be the primary addictive component in tobacco, and yet, as much as we do know, the neurobiology is still incompletely understood. So with that, I want to talk a little bit about neural circuitry generally in substance use disorder. So this is a pretty busy slide, but I really am just going to zero in on one main pathway. So I really focus a lot on the nucleus accumbens core, where that yellow star is. And I'm also going to be talking about this glutamatergic projection from the prelimbic subregion of the prefrontal cortex, where that green arrow is pointing. So neuroimmune mechanisms of substance use disorder. What do we know across drug classes? Well, just to zero in a little bit on microglia, they express receptors in the mesocortical limbic reward pathway, and these have been implicated in addiction processes. For example, toll-like receptors, or TLR4 specifically. Further, Kim Nixon's lab recently found an increase in phagocytic ability of microglia after alcohol. As well, opioids and cocaine have been associated with toll-like receptor activation within the nucleus accumbens. So we know across drug classes, there seem to be an involvement in microglia and neuroimmune mechanisms. So just to tell you a little bit about microglia, these are the resident immune cell of the brain and are activated following drug use. And this was something we covered in a recent review paper we had this year. They are phagocytes, and in a healthy brain, they clear out apoptotic cells and proteins such as amyloid beta, which are known to contribute to neurodegeneration. They are also involved in the remodeling of neuronal connectivity. And when activated, they have these processes which retract, and then they take on this amoeboid-like structure. So keeping all that in mind as I go through the data that I'm going to show you next. Okay, so what about neuroimmune mechanisms and specifically microglia and nicotine use motivation? Before I get to that, you may be wondering, is nicotine pro or anti-inflammatory? Well, it's a bit confusing. In the literature, it's thought to act through nicotinic acetylcholine receptors to decrease immune response in Parkinson's disease. Yet, contradictory results are found in Alzheimer's disease. Further, some studies show that nicotine actually suppresses macrophages and reduces mortality in conditions such as sepsis. And other studies show that cigarette smoking exacerbates multiple sclerosis and Crohn's disease. So as you can see within the literature, there seems to be evidence for both pro and anti-inflammatory mechanisms of nicotine. But we're really more interested in chronic nicotine exposure, and there is evidence that this may produce a pro-inflammatory state. So studies in the literature show that chronic nicotine exposure increases blood-brain barrier permeability, oxidative stress, and brain infarct size in post-ischemic inflammatory response. Within chronic nicotine exposure itself, one study found that microglia actually exhibit morphological changes following chronic nicotine exposure via an osmotic mini pump and during withdrawal. So again, I'm asking, what about neuroimmune responses in the reward pathway following chronic volitional nicotine consumption? So this is a pretty heavy figure here, and I'm going to kind of go through it step by step. But before we can really get into these neuroimmune mechanisms, it's really important that first we understand glutamatergic signaling, because these things do interact. So I'm going to come back to this figure. So first, understanding glutamate signaling in nicotine addiction. So again, if you just look at that sagittal slice of that brain, I'm focusing on that prelimbic glutamatergic afferent into the nucleus accumbens. And here we have, these cells synapse onto medium spiny neurons within the nucleus accumbens and signal. And then on astrocytes, there's this blue protein you can see, and that is the glial glutamate transporter, GLT1. The other protein there is a cysteine glutamate exchanger. So during drug-seeking behavior, several studies across drug classes have found cue-induced glutamate release during reinstatement of drug seeking, including nicotine, from these prelimbic glutamatergic afferents. This glutamate is then released and leads to transient synaptic plasticity, which I will show you in a minute, in these medium spiny neurons. And also, there's a downregulation of GLT1 in astrocytes, which leads to this dysregulated circuit. Okay, so how did we figure any of this out? We can actually use rodent models to reveal the neurobiology of nicotine use motivation. So in my lab, we use a rodent model or rat model of nicotine self-administration and reinstatement. So you can see here a graph that shows lever presses, and the rat is basically, we put in a jugular catheter into their jugular vein, and following recovery, we teach them if you press this lever, nicotine will be delivered intravenously and paired with a light and a tone compound stimulus cue. And it's always contingent upon that lever press. There's also an inactive lever to control for non-specific responding. Rats will acquire nicotine self-administration and eventually hit a kind of a stability during maintenance. Following self-administration, we can then put them into something called extinction, where now lever presses lead to no program consequence. And we'll typically see this extinction burst where lever pressing will increase at first, and then as they learn that the primary reinforcer and the stimuli are not being delivered contingently upon a lever press, we see this decline in lever press behavior across the extinction phase. Then what we can do is use these priming stimuli to basically induce active lever pressing in these animals. So we can either give them back those contingent cues previously paired with nicotine, we can give them a drug priming injection. People also use stressors such as foot shock to basically induce resumption of active lever press behavior, and that is reinstatement. Okay, so I'm going to focus first on this prelimbic glutamate release mechanism within the nucleus accumbens core, because this had been shown in the Clivus lab with cocaine seeking and heroin seeking, but I came in and wanted to know if this is also happening in nicotine seeking behavior. So using in vivo microdialysis, I basically train the animals up on nicotine self-administration and extinction. And then during reinstatement, I was actually measuring extracellular glutamate within the nucleus accumbens core. So you can see here active lever pressing is very high in the beginning of the session, and these are 20-minute bins. And then as the rats start to learn that those cues aren't being paired with nicotine itself, we tend to see this drop off in active lever pressing across the 2-hour session. Now, if you look at the glutamate data, you can see after the first 2 hours of just measuring basal glutamate between nicotine and saline animals, that arrow there indicates the levers are coming out. And as they get these contingent cues previously paired with nicotine, we see this increase in extracellular glutamate early on in the session, and then also it declines across the 2 hours. Okay, so now I'm going to focus on that postsynaptic cell, those medium spiny neurons. So we can actually measure this both structurally and functionally. We basically can measure dendritic spine morphology. We can fill the cells with lipophilic dye, and in this case, I always use dye I. But we can take different measures from this as outputs of long-term potentiation and long-term depression to provide measures of changes in synaptic strength. In other words, increased spine head diameter, increase in density. We can also look at neck length, all of these different measures to get readouts of structural plasticity. So the bigger the spine head, we consider that potentiation, and the smaller the spine head, we consider that depression. Functionally, we can also use whole cell patch clamp electrophysiology to measure glutamate currents. We can isolate AMPA and NMDA currents, and we basically take a ratio between the peaks of those currents to get our functional readout of AMPA to NMDA ratio. And so here you can see data from a paper I had a few years back during my postdoc years. Here you can see a dendrite that we have imaged using confocal microscopy, and using Imaris, we can actually measure all of these different aspects of spine morphology in 3 dimensions. And so here you can see this graph where I've got yoked saline animals. I've got a T, or time equals 0 time point, which means animals went through nicotine self-administration followed by extinction in that red bar. And as you can see, if you compare the red bar to the white bar with spine head diameter, there's kind of a basal increase in spine head, meaning that these cells during withdrawal seem to be resting in a potentiated state. When animals are given a 15-minute time point with those contingent cues, remember that's when glutamate is higher, we see this rapid increase in spine head diameter, which then collapses back down at the 45-minute time point, along with that kind of decline in glutamate. And similarly, when we look at AMPA to NMDA ratio, we see an increase in that red bar compared to the white bar, which is withdrawal from nicotine with extinction. We see this basal potentiation, and within 15 minutes of getting that cue, we see this further potentiation of the ratio. So we concluded that cue-induced nicotine seeking induces rapid transient increases in spine head diameter and AMPA to NMDA ratio in the nucleus accumbens core. Okay, so now I want to switch gears and focus on GLT1 in the astrocyte. And this protein is responsible for uptake of about 90% of glutamate that's released. And so I'm really focusing here because a lot of drugs are targeting this protein currently in clinical trials. So I was really curious if this protein was downregulated after withdrawal from nicotine. So I ran a panel of Western blots on AMPA receptors, NMDA receptors, and glutamate transporters. So if you focus just on GLT1 here for this purpose, you can see the black bar, or the nicotine group. I found a decrease in GLT1 protein expression compared to saline animals. So I did see this protein expression that was downregulated. I was really interested in this 15-minute time point because of all these other effects that we were seeing. And so when I ran the same Western blot with the 15-minute time point, we saw protein expression actually rapidly increase at that 15-minute time point. So we concluded that GLT1 is downregulated following withdrawal with extinction from nicotine, and then is rapidly upregulated following 15 minutes of reinstatement. And if you're wondering if this is functionally relevant, in collaboration with Sade Spencer and Coty Garcia Keller, we ran an uptake assay and found that compared to saline animals, we did see this decrease in sodium-dependent uptake of glutamate, which is through GLT1. Conversely, sodium-independent uptake, or through the cysteine glutamate exchanger, was not different between these two groups. So we concluded that GLT1 function is also decreased following withdrawal from nicotine self-administration. Okay, so is upregulation of GLT1 a good treatment option for nicotine use disorder? One compound that's been looked at in clinical trials is N-acetylcysteine. This is FDA approved, it's well tolerated, it's in clinical trials for substance use disorder, including smoking, and in preclinical models, it upregulates that GLT1 protein. And a few years back, Kate Reisner had a paper showing that GLT1 upregulation is necessary for NAC to prevent cocaine-seeking behavior. So I came in and asked, what about nicotine? And so a master's student in my lab a few years ago ran a similar study to Kate's, where we were holding down GLT1 with an antisense vivo morpholino, and our hypothesis was that doing this would block NAC's ability to decrease nicotine seeking. So in other words, if you give NAC, you will upregulate GLT1 and reduce reinstatement. Conversely, if you give NAC with that antisense vivo morpholino, NAC can't restore GLT1, and therefore reinstatement will occur. And that's what we did see. So this reinstatement data graph here shows extinction, active lever pressing compared to reinstatement, active lever pressing. And just to orient you to the groups, we had control morpholino NAC or saline in black or white, respectively, and then antisense vivo morpholino NAC or saline in gray and blue. And you can see that the only group that did not reinstate is the control morpholino NAC group. All other groups reinstated. And when we looked at GLT1 expression, GLT1 was down in all of those groups compared to that control NAC group. So we concluded that NAC attenuates cue-induced nicotine seeking through this GLT-1 dependent mechanism akin to cue-induced cocaine seeking. So in other words, chronic NAC increases GLT-1, blocks reinstatement. GLT-1 with an antisense morpholino plus NAC, GLT-1 cannot be restored and reinstatement occurs. Okay, so how exactly is NAC working to decrease nicotine-seeking motivation? Well, NAC actually activates the cysteine glutamate exchanger. So it was kind of this molecular black box with a question mark on it for us about how it is then going to upregulate GLT-1. So how is it doing that? Well, first, let me give you a little bit of background on the NF-κB pathway. So what happens is you have these extracellular signals, such as the pro-inflammatory cytokine TNF-alpha. This then binds to its receptor, activating it. This then activates I-kappa kinase, and through phosphorylation, ubiquitination, and degradation of I-kappa B, NF-kappa B is then translocated into the nucleus, and then this leads to gene expression or repression of various gene targets, including EAAT2, which codes for the protein GLT-1. This also targets TNF-alpha itself, IL-6, as well as CD40, which is a microglial marker that I'll get back to. Okay, so knowing this, we have this hypothesis that there are interactions between neuroimmune mechanisms, NAC, and nicotine-seeking behavior, because NAC inhibits TNF-alpha-induced NF-kappa B activation through inhibiting I-kappa kinase. So is NAC actually working as an anti-inflammatory to decrease nicotine-seeking behavior? Well, we asked Scott Russo for some viral constructs that he generously donated to us, which allowed us viral-mediated gene transfer to express either a constitutively active or dominant negative mutant of I-kappa kinase. So this was IKK constitutively active or dominant negative into the nucleus accumbens core. And just to clarify, the constitutively active form activates NF-kappa B and the dominant negative form inhibits it. So Mark ran this study as part of his master's thesis. This is the general timeline of experimental procedures, very similar to what I've described already. Animals underwent surgery and recovery, and then underwent nicotine self-administration at a 0.02 milligram per kilogram per infusion dose, followed by that extinction phase. And towards the end of extinction, we intracranially administered these herpes simplex viruses, which we also validated, either the dominant negative, the constitutively active, or the control virus, concurrently with 5 days of NAC injections, followed by reinstatement and brain tissue collection. Okay, so just to orient you to this graph, again, it's extinction and reinstatement active lever pressing, and we've kind of color-coded this according to viruses. So our control virus or GFP virus is in gray, and either animals receive vehicle or NAC. The dominant negative virus is in green, and again, either vehicle or NAC. And in red, we have the constitutively active virus with vehicle or NAC. And as you can see, just going through the gray bars, these animals did reinstate when given vehicle, but NAC occluded reinstatement in this group. Conversely, the dominant negative groups, we saw no reinstatement, either vehicle or NAC. And in this red bar group, the constitutively active, now this is where things got interesting. When we activate I-kappa kinase, it occluded NAC's ability to reduce nicotine-seeking behavior. And so we concluded that the NF-kappa B pathway mediates conditioned nicotine seeking as well as the inhibitory effects of NAC. And when we checked GLT-1 expression, we collapsed across NAC condition just to look at the viruses. And what we found was that the dominant negative inhibited reinstatement regardless of NAC treatment, but NAC did not increase GLT-1 in this group. So we concluded that the NF-kappa B pathway is having effects independent of GLT-1 expression. And so getting back to this question, is targeting GLT-1 sufficient to reduce drug-seeking behavior? Well, it's important to note that NF-kappa B is a bidirectional regulator of GLT-1. Thus, both activation and inhibition of this pathway can repress GLT-1 transcription, which kind of makes sense with our data. Also, Carly Logan, a few years ago in the NACSTUD lab, found that specific rescue of accumbens GLT-1 is not sufficient to attenuate cocaine-seeking behavior. So again, we started asking ourselves, are there GLT-1 independent mechanisms, in other words, immunomodulation, that may underlie efficacy of drugs such as NAC? Well, we also wanted to check that pro-inflammatory cytokine TNF-alpha. Here we show that NAC reduced nicotine reinstatement-induced release of this pro-inflammatory cytokine, and we measured that soluble released form. And here you can see, compared to saline-treated animals, NAC reduced TNF expression. We also wanted to check astroglia because this glial fibrillary acidic protein, GFAP, is generally accepted as a marker of astroglial activation, and we're really focusing on astroglial contributions to the circuit. Also, Michael Schofield and Kate Reisner with Peter Clivis found that GFAP is decreased in the accumbens score after cocaine. So we checked it in our animals, and we found that after nicotine withdrawal and reinstatement, it is also downregulated, but this is not reinstatement dependent. And so we have plans going forward of looking at, astrocytic contacts with neurons and microglia, because now we're showing that GFAP is down. So that's another future direction in collaboration with Michael Schofield. Okay, so I want to flip a little bit to microglia and nicotine. I mentioned CD40 being regulated by NFWB. This is required for microglial activation. So we hypothesize that it's upregulated after nicotine. And so Mark checked this with our antisense and sense animals. And what we found is when we suppressed GLT-1, we increased immune activation after reinstatement. So again, this is after the animals reinstated. So we concluded that the GLT-1 vivomorpholino activated microglia after reinstatement, regardless of NAC administration. And so we thought that these data actually show that nicotine-seeking behavior activates microglia. Okay, so I just want to go through that busy picture with you. This was something in collaboration with Michael that we created for that paper. We have this kind of hypothesized glutamate neuroimmune mechanisms that interact within the reward pathway. So I haven't talked too much about TLRs, and I have plans to study them, but specifically, we think drugs of abuse are activating toll-like receptors, which trigger this NF-kappa B signaling pathway within microglial cells. They then release pro-inflammatory cytokines, such as TNF-alpha and IL-1 beta, and these cytokines then bind to their receptors on astroglia, which activates NF-kappa B. This then leads to repression of GLT-1 transcription and ultimately downregulation of the GLT-1 transporter, because TNF-alpha negatively regulates EAAT2 transcription and therefore GLT-1 protein. This results in an inability of astroglia to then clear excess glutamate released during cue-induced reinstated nicotine-seeking or drug-seeking behavior. And following exposure to these drug-associated cues, glutamate release from cortical afferents into the nucleus accumbens is potentiated, leading to activation of ionotropic glutamate receptors such as AMPA and NMDA, inducing rapid transient postsynaptic plasticity and subsequent relapse. Okay, so far we've shown a marker of microglial activation and pro-inflammatory cytokine expression is increased following nicotine seeking. But what about structural changes in microglia following nicotine consumption? So we can actually measure microglial activation through studying structural changes. So ramified microglia become activated following acute injury, which I already kind of described in the beginning, but here you can see in this image, ramified microglia have these long processes, and then when they become reactive, those processes retract and cell bodies enlarge, and there's hypertrophy due to acute injury. And as we published this review, we noted that activation actually lies on a spectrum, and it's not binary. It's not either activated or not activated. And so it's really important to measure microglial morphology with precision to get more at exactly where these microglia are in terms of activation. There's different ways to do this, including 2-dimensional versus 3-dimensional and manual versus automated. So this is one way to do it. This is the manual 2D analysis of microglia. So as you can see here, there are ramified microglia on the top after saline. And as I'll show you more, we have activated microglia after prolonged nicotine exposure here on the bottom panel. Now, in order to do this manual analysis, this image is converted to binary and background. So anything not attached to the cell is then omitted. This is converted to the skeleton image that you see on the right. And now you can measure endpoints, process length, circularity, etc. But tracing of cells using 2D analysis ignores 3D information and is subject to bias. And so you can also do this with a automated 3D analysis or manual 3D analysis. Imaris is a program that you can use to do semi-automated analysis, but this is time-consuming, expensive, and potentially subject to bias. Even with spine morphological measurements using Imaris, we have to kind of go in and tell Imaris what is a spine and what isn't a spine. So anytime an experimenter has to go in and kind of manipulate that, it can be subject to bias or experimenter reliability. So Emma Bondi came into my lab and she found this paper by another group, York et al. In 2018, published this in eNeuro. It's something called 3D Morph. It uses GitHub open source code. It removes user bias and increases reproducibility. So she got this working because she's amazing. You can see differences here already probably between the saline and the nicotine animals, but after taking these high resolution 3D images, the microglial stacks are first converted prior to eliminating any non-microglial cell debris. And then these stacks are converted to find the cellsomas prior to analysis. And you can see that this is kind of color coding the different parts of these cells. And then after she kind of got this working, we wanted to know exactly how different 3D Morph was from this manual analysis she had been doing. And what we found here was just looking at maximum branch length, we found that 3D Morph, or this automated analysis, detects more features of cellular structure than the manual analysis. So we were very excited to see that. And first, she ran a cohort of female animals, either nicotine or saline self-administration, and she found some really interesting results using this automated analysis. These different measures that you see here are all measures that can tell you whether microglia are activated. So percent occupied volume, number of endpoints, and territorial volume were all decreased in our nicotine self-administering females compared to saline. And so we concluded that microglia are structurally activated following nicotine in these animals. She also did vaginal swabs in these females, and she is going to start looking at her data according to estrous cycle phase to determine if that changes microglial activation. Okay, so just to show you these 3D images that were being analyzed, here's a saline video, and we'll play the other one. Well, the nicotine video is right here. There we go. And you can see pretty noticeable structural differences. And so we concluded that microglia appear activated following nicotine self-administration compared to saline. Okay, so now I want to get in a little bit to sex differences because that's a near and dear to my heart topic that we've really been focusing on in the lab as well, and that may be relevant to neuroimmune signaling. So women clinically it's been found that they are typically more vulnerable to substance use disorders, and long-term smoking cessation is more difficult to achieve in women than men. Further, women are less likely to quit smoking than men in clinical trials for pharmacotherapies, including the patch. And in women, ovarian hormones that cycle during the menstrual cycle are associated with nicotine vulnerability and resilience, such that estrogen or E2, 17-beta estradiol or E2, is associated with nicotine vulnerability, and progesterone is associated with resilience. Further, there are sex-specific differences in microglial activation and neuroimmune signaling, which may contribute to greater susceptibility of females to psychological disorders, including addiction. So then we asked, what about sex-specific neuroimmune mechanisms in nicotine use in our model? So Emma next ran males and females and compared their nicotine consumption as well as microglial activation. And here you can see if we just take all of their 15 sessions of acquisition and we combine their nicotine consumption to take an average, we see that males generally consume more nicotine at this 0.06 milligram per kilogram per infusion dose than females. However, what we found when we look at the mean ramification index of microglia we see that it is higher in males. And you can see here that female microglia appear to be more activated. So we concluded that although males consume more nicotine than females, they show less activation of microglia, or in other words, higher ramification. So these results show that females may be more susceptible to microglial activation following nicotine self-administration. Okay, so now I'm going to flip a little bit. I've given you some mechanistic results showing that neuroimmune signaling is involved with nicotine motivation, but can we use this information pharmacotherapeutically to treat tobacco use disorder? Well, first, I want to go back to N-acetylcysteine because I've talked a lot about it. A colleague and friend of mine, Dr. Erin McClure, ran a clinical study with NAC and smoking in both women and men. And this is, again, a currently available glutamate compound, but it has checkered clinical success, including for smoking. So this paper just came out. We collaborated on this. It's out in Drug and Alcohol Dependence in Press. And you can see this arrow is pointing to either NAC or placebo with time to relapse in days on the x-axis. And you can see that placebo-treated individuals actually did better in terms of time to relapse than the NAC group. So we concluded that NAC was not efficacious in reducing relapse to smoking. So leaving NAC aside, there are other compounds that may have better efficacy. So we kind of switch gears to Troraluzol. This is a pharmacokinetically and metabolically enhanced analog of Raluzol, which is currently FDA approved and used to treat ALS. This compound may have anti-inflammatory properties. As well, Kate Reisner's lab showed a few years ago that Raluzol has been shown to decrease cocaine-seeking behavior. So there's some feasibility that a similar compound, Troraluzol, may be efficacious here. So we are asking about nicotine seeking in collaboration with Dr. Scott Rawls. And also we have a collaboration with Bill Stoops to maybe try to get this compound into a clinical lab study. Okay, so Troraluzol was developed by Biohaven Pharmaceuticals. It increases glutamate uptake from the synapse and augments expression and function of GLT1. It's currently being evaluated in phase 2 to 3 clinical trials for Alzheimer's, OCD, ALS, and spinocerebellar ataxia. It's also well tolerated, and Scott has shown that it has promise in reducing cocaine and opioid seeking behavior in his models. So on the website, Biohaven shows that it has improved bioavailability, no negative food effects, lower overall drug burden on the liver, optimized dosing regimen and compliance, and the potential for developing multiple formulations. And so we asked, does this work in nicotine? So we, again, these animals went through a similar behavioral paradigm as I've shown you before, surgery followed by either saline or nicotine self-administration at the 0.02 milligram per kilogram per infusion dose, followed by extinction. And during extinction, animals got either daily Troraluzol injections at the 8 milligram per kilogram dose IP or vehicle. And then following extinction, they received reinstatement, and then we collected brain tissue. And I should note, this is all in male rats. And what we found, these are preliminary data, but they suggest that chronic Troraluzol may actually facilitate extinction learning when given daily following nicotine self-administration. So in this graph, you can see the filled in circles, that the black line is Troraluzol and the green line is vehicle. And you can see lower lever pressing across sessions in the Troraluzol group. Further, when we looked at, this is not that surprising. It's similar to NAC in male rats. So this was a paper we had a few years ago. A postdoc in my lab, Greg Powell, published these data showing that chronic NAC during extinction did a similar thing where it facilitated extinction learning and blocked reinstatement. So when we look at reinstatement in these animals, we show that this dose decreases reinstatement in our model, but we may need a higher dose because it didn't completely block it. So we're hopeful that this may be the beginning of a possible therapeutic for smoking. So the next thing we did was we ran a cytokine and chemokine analysis on brains from these animals. So it was a 2x2, right? Troraluzol, vehicle, nicotine, or saline. And I'm going to focus specifically on our results from the nucleus accumbens for this talk. So here you can see, first we looked at CXCL1, which is a chemokine that recruits and activates neutrophils for microbial killing at the tissue site. We found that nicotine itself in our vehicle-treated animals didn't elevate CXCL1 in the accumbens core, but when you give Troraluzol, it upregulated CXCL1 only in nicotine animals. So that was a very interesting result. The next thing we wanted to look at, though, of course, was TNF-alpha. So this is membrane-bound TNF-alpha that was measured here, and we found in our vehicle-treated animals, it was down-regulated by nicotine. Presumably, and again, these brains were collected after reinstatement, so activity had already happened. So we are hypothesizing that this downregulation is due to TNF being cleaved and released during reinstatement, and that's why we see that it's down. But when you give TRLZ, it normalized this, indicating that less TNF alpha may have been released during reinstatement. So we have a lot of plans to kind of further delve into the neural mechanisms of this. Okay, so this is our hypothesized mechanism of TRLZ, some of which I've shown you, but there's a lot more work to be done. So first I want to talk about microglia. We think that it may decrease release of IL-6, TNF-alpha, CXCL12, CXCL1 from microglia. There's also reason to think that it decreases glutamate release presynaptically in those prelimbic glutamate afferents. We also think that it can indirectly perhaps increase glutamate uptake through GLT1. And subsequently, a downstream consequence may be reducing AMPA to NMDA ratio and that transient plasticity in those medium spiny neurons. But this is all stuff we have to do in the future. So our future directions, we have several ongoing, one of which is to look at these hormone neuroimmune interactions in nicotine use motivation. So this very complicated schematic here shows you how estrogen can actually influence NF-κB signaling. So that's really interesting. So actually, we have found that ovarian hormones regulate nicotine consumption as well as glutamate plasticity after nicotine. So here you can see when we ovaryectomize female rats, so we're taking their ovaries out, this decreases nicotine consumption. As well, OVEX increases AMPA to NMDA ratio after immediately following nicotine self-administration. So these animals are not in withdrawal. So that's looking at that purple bar compared to the black bar. And those black bar animals are ovary intact. Next, when we give E2 supplementation, we actually see a reversal of that decline in intake. And we also see a reversal of this increase in AMPA to NMDA ratio by E2. Interestingly, this is nicotine specific because when you look at ovaryectomized female rats that did not undergo nicotine self-administration, we don't see that increase in AMPA to NMDA ratio. So we have a lot of work left to do, but obviously there's biological feasibility that hormones are playing a huge role here in potential sex-specific effects. So future directions, my postdoc, Dr. Erin Mayer, is really interested in studying estrogen receptor contributions to neuroimmune signaling in females during nicotine consumption and seeking across the estrous cycle. And you can see a beautiful image that she took on the confocal looking at estrogen receptor localization. This is specifically in the ventral tegmental area on dopamine neurons, but she's also got data to show localization of these receptors in the nucleus accumbens. This is interesting. Again, I'm just going to say E2 is associated with smoking vulnerability in women. And it can also modulate neuroimmune signaling through the NF-κB pathway via estrogen receptor beta. So we have a lot of plans to look at this receptor particularly. So our question is it possible that NF-κB signaling underlying nicotine seeking is different in females as compared to males? We're asking this question because all of the data I've shown you thus far that we've collected on the NF-κB pathway was in male rats. So we haven't yet figured out if this is different in females. The next thing we're doing is we have this goal to actually control microglial activation, but this has been kind of difficult in the field because infecting these cells, which are immune cells, is kind of difficult. So we're actually trying to use chemogenetics to activate or inhibit microglia via the CX3CR1 pre-transgenic rat line. So just to go through this panel, this is accumbens tissue. You can see the anterior commissure is noted in white, in a white line. Here you can see IBA1, which is our microglial marker in green. We have CRE in red in that middle panel. And then when we merge them, we see that Cre is actually in microglia. So check, but next we have to validate that our viruses are getting in there. So that's next on the docket. So just some other ongoing studies that Emma is currently doing. We're using CX3CR1 to try to kind of get drugs into those cells, but we're actually interested in the receptor itself and its ligand, CX3CL1, because this is expressed on microglia and may actually influence adenosine signaling, which we're also interested in looking at. So Emma's actually currently running a group of animals to look at the adenosine A2A receptor and how it's contributing to the circuit I've been describing. Down the line, we also have plans to look at adenosine signaling itself. Okay, so just to summarize, neuroimmune signaling may be a critical driver in nicotine use vulnerability, and targeting neuroimmune signaling may yield promising clinical outcomes to promote smoking cessation. But more research is needed to determine sex differences in neuroimmune signaling and nicotine use. So with that, I'd like to thank you all for being here. I have an amazing lab, past and present. Currently, my postdocs are Erin Mayer and Shailesh Khatri. My research technicians are Emma Bondi and Mei Hong. And of course, I want to thank the undergraduate volunteers. None of this is possible without all of them, and they're amazing. I also want to thank my collaborators, Drs. Scott Rawls, Josh Beckman, who helps with a lot of the statistical analyses, Aaron McClure, Michael Schofield, Bill Stoops, and Heather Bamonte Nelson, who helps with all of our hormone stuff. We're also funded by NIDA, that we've received generous money from the Arizona Alzheimer's Consortium, as well as the Substance Use Priority Research Area at the University of Kentucky. And Shailesh and I just received a pilot grant from them as well, so we're very grateful for their support. And thanks. Should I stop sharing?
Yeah, I was going to say, if people have questions that might be more specifically answered with the slides, we can always pull them back up.
Okay.
Great. So, Ankush, do you want to start with the question in the chat? And now is a good time if people have more questions to drop them in the chat, and we'll try to get through them.
Yeah. So Aaron McClure says, excellent presentation, Dr. Gipson-Reichardt. Given that TLRZ has promising preclinical data across several different substances, would you therefore would there be promise in testing the compound for polysubstance use in humans?
That is a wonderful question. First of all, hi, Aaron. And second of all, we are actually looking at that. We're submitting a grant right now to look at nicotine and alcohol co-use. There's some justification to think that this compound may work in both, but it's, again, not known. And developing polysubstance use models in rodents can be challenging. So unfortunately, not unfortunately, but we study animal, in our animal models, we study drugs of abuse in kind of isolation. And there's a benefit to that in that we can isolate drug-specific effects, but that doesn't really represent the clinical experience, right? Because most people are polysubstance users. So I think it's really an important endeavor. It's something that I'm actually collaborating with Bill Stoops right now. We have a grant to do oxycodone and cocaine co-use and looking at NAF as a possible treatment. But again, it's a wide open area.
Okay. So,
oh, sorry.
Yeah, go on.
Oh, no, I'm done.
Sure. Marina Picciotto says, great talk, lovely data. What types of NAcetylcysteine
Nicotinic receptors,
do you think might be regulating microglial structure?
So I've thought a lot about this. The alpha-7 receptor actually, it's pretty ubiquitous. It is calcium permeable. It regulates glutamate release, although I've never seen a paper showing that in the accumbens. But I've often thought about it in terms of its ability to modulate microglia because it's also expressed on them. That's not something I've done, but I would love to look into that going forward.
I have a question. So you showed microglial NF-kappa B activation through TNF-alpha and IL-1 beta could inhibit GLT expression, right? So TNF-alpha and IL-1 beta have also been implicated in treatment-resistant depression. a lot of patients show higher levels of those cytokines. And do you think there is an aspect of stress contributing to that?
Most likely. I don't study stress specifically in my lab, but there are several papers out there looking at stress and neuroimmune activation, especially microglia. And there may be sex-specific and brain region-specific effects of stress on different cytokines and microglia. Also, isolating depression from smoking, a lot of times there might be overlap. And so, that's a really interesting question. I wonder if there isn't a synergy there if someone does have both depression and nicotine use disorder. So I think that's a really wonderful question that needs to be answered.
I have one more question, which is there a difference? So you showed how microglial activation was different in males and females, right? So is there a difference how males and females perceive that nicotine high? And is it more rewarding to one sex more than other, something like that?
There are clinical data showing, that males and female, or women and men, respond differently to different doses of nicotine, have different preferences for high or low doses. So there are sex differences in, nicotine, preference and smoking patterns. So I'm sorry, can you repeat your exact question? I got lost in my thought.
So my question was the way that males and females or men and women perceive the nicotine high is different. Is it more rewarding to one of the sexes?
So like I said, there is clinical literature to suggest that women have a harder time quitting. And again, like I mentioned, there are differences in preference for different doses. I've been actually particularly interested in women undergo very specific like reproductive life changes, right? So there's throughout adulthood we undergo menopause, and actually there are data to suggest that menopausal symptom severity is associated with difficulty quitting as well. So there's all these little bits of evidence to suggest that women have a very different experience. And not to say that men don't undergo andropause across the lifespan, because that is kind of their trajectory. But I'm really interested in looking at that, and I do think there's clinical and preclinical evidence to suggest that there are differences in nicotine use and smoking patterns.
I had one quick question from just before it's a little bit, going back to maybe bridging some of the work that you did in your postdoc, where you talk a lot about, the MPFC, or the infralimbic projection, and glutamate coming into the accumbens from that source. So what, do you think this is a synapse-specific mechanism that the, astrocytic glutamate clearance is working on, or do you think it's going to be relevant to the different excitatory inputs coming into the accumbens, or is there something about the organization of the MPFC synapse that's particularly relevant for this astrocytic, controlling the global tone of glutamate?
I think you raise a really good point. I know that Peter's lab has kind of looked at the different circuits coming in, such as from the basolateral amygdala, for example. And you're right, glutamate's coming in from all kinds of different places, and I'm just isolating this one pathway. And so I have never looked at all of these mechanisms in terms of other neural inputs, but I think that that's a really important point. And others have done some of that work, but in terms of what I've done, I can't say for sure whether other glutamate sources are contributing to these effects. But it's a great point.
Is there, and to be honest, I know nothing about how astrocytes work. I tend to ignore them, but so I realize we're recording now. Oops. But yeah, just wondering if there's something, do the astrocytes organize themselves in a synapse specific way, or even in relation to the medium spiny neurons, like closer to the cell bodies or out in the dendrites, or is there really not that kind of organization?
Yeah, so they're large and they ensheath synapses. So they actually kind of strategically are placed where GLT1 is near that synaptic cleft. And so Peter's lab and Michael Schofield and Kate Reisner have shown that there's retraction of the astrocyte from that synapse after withdrawal from cocaine and I think heroin now. And so it seems that that's part of the dysregulation of astrocytes, is that they're not where they should be. And so we have this hypothesis that there's, we saw that rapid increase in GLT1 at that 15-minute time point. And so I'm kind of thinking that it's similar to Peter's. I think he just had a paper showing that astrocytes are returning to the synapse during that early part of reinstatement. And it kind of goes in line with that GLT1 increase. But we have a hypothesis that that's not, that it's a transient increase, because astrocytes are so dysregulated and NFWB signaling may be, playing a part here in repressing further GLT1 transcription. So even though astrocytes might be trying to get back there, putting more GLT1 there to try to clear that glutamate, it's kind of like, it's so dysregulated that it can't do an efficient job of it. So we have to further look at that, but that's kind of how we think about, what's going on.
Great, thank you.
So I have a related question, which is, other than glutamate, does microglia in the nucleus accumbens also have receptors for dopamine, serotonin?
Oh, yeah. So I'm trying to think specifically which ones, but there are, roles for those other neurotransmitters in immune activation, both in the periphery and in the central nervous system. And so I'd have to look at specifically what they're expressing, but I'm pretty sure that they do, but don't hold me to that. I have to look at it.
Sure. We have another question from Erin who asked, any data on Troraluzol for cannabis?
No, this is, so the only data we have so far is preclinical, and the clinical data with this compound is really not in substance use disorder. So not yet that I know of.
And I believe there is one sort of half unresolved question in the chat.
Okay.
Which was, again, from Erin McClure. She thanked you for your answer. And any data on Troraluzol for cannabis use? Sorry.
Oh, I think I just answered that one, right?
Sorry.
Okay, I guess that's it for today.
Well, thank you all for everything. I enjoyed it.
We did too.